Hey everyone, Savvy Amber. Today I'm going to talk about a way to culture bacteria. You may be wondering how scientists are able to study bacteria since they're so small and microscopic. There's different ways to study them or to grow them. I'm going to be talking about the streak plate method, and this involves growing them in a petri dish, which most of you are probably familiar with from your science classes. So what do the bacteria need, and how do you grow them in a petri dish? Well, one thing they need is nutrients, just like us. They need stuff to eat. So we put a nutrient in the petri dish that contains all the nutrients they need, plus agar, which makes it jello-like. So it's kind of picture a bowl full of jello for to picture the consistency. I'm going to show you the step-by-step process for making a streak plate. The basis of it is to do 4 quadrants of streaking on that jelly-like surface of the agar. My shirt actually shows the method right here, and my microbiologist loved a streak t-shirt, one of my favorites. But you can see the 4 different quadrants on the shirt, and then one of our favorite tools of microbiology, the inoculating loop, which is used to pick up and transfer cultures. But in the video, demonstrating the step-by-step process, I'm going to be showing transferring a culture from a plate. So the first thing here is sterilizing the inoculating loop. This is to prevent contamination, which is very important in doing transfers. And here I'm selecting a colony off of the plate for transfer, a nice slimy one. And the plate that I'm transferring to is being kept upside down. That just keeps other bacterial cells from being able to fall on it. That's another way to keep unwanted bacterial cells off of it. Because what I'm looking into is what I'm putting on the plate. So I just kind of gently swiped that loop across the surface of the plate. Now the loop is being sterilized again. It's important to do this between each step. It removes the bacteria on there, prevents contamination. Now I'm getting ready to do the second quadrant. You can see the first one, kind of where it is, because it leaves a little bit of an impression in the agar. For the second one, you just drag it through a little bit of that first one to pick up some of those cells and kind of thin them out. Sterilizing the loop again and getting ready for that third quadrant streak. And by this third one, we will be diluting the cells quite a bit by this point. You can repeat the process we did with that first quadrant. And if you see that little tap on the agar with the loop, that's just a way to kind of help cool it down, just to make sure it's cool before running it back through the culture again. It helps dissipate the heat. There's the third culture, or third quadrant. Sterilize the inoculating loop again. And you can see where all three quadrants are. So for fourth, kind of do the same as before. Run the loop through, pick up a few of the cells. And that plate is complete and ready to go into the incubator. That will be incubated for 24 hours. And then after 24 hours, you'll be able to see the bacterial growth on it. I will show you what the plate looks like after 24 hours. And if you take a look at this plate, you can see the first quadrant. It's just completely covered. It's like a carpet of bacteria. And then by the time you get to the second quadrant, it's still pretty covered. You're starting to see a few individual circles on there. By the third, you can see several individuals. Sometimes it works like this, where you get the separate colonies in the third quadrant, and the growth just doesn't make it to the fourth. And that's fine if that happens. That your main point is just to get those isolated colonies, because now that you have those, let's say in this sample I was trying to see how many types of bacteria I had. I wanted to know if I had a mixture. Here I can see there is a mixture. As you can see, the small colonies, there's some bigger ones. We've got a couple different colors going on there, so there's a mixture. That sums up how to make a streak plate. If you have any questions for me, please drop them in the comments below. And if you like this video, please like, subscribe, and I'll see you next time.